Welcome to This Explainer. Today, we're diving into some incredibly fascinating source material that outlines a literal, DARPA-level healing ray gun. It uses intersecting fields of physics to regenerate tissue, sounding way less like a hospital and a whole lot more like something pulled straight out of a sci-fi video game. They call it the Sono-Photon Bioregenerative Projector. It's an amazing concept that merges photonics, acoustics, and electromagnetism to create what researchers actually refer to as a quantized ball of healing.
Okay, so what if you could regrow a human limb just by shining light and sound at it? The source material we are analyzing today absolutely isn't magic. It's a highly detailed blueprint for a completely non-invasive device that triggers your own cells to rebuild themselves. We're talking about leveraging FDA-cleared dosages of physics to do something that honestly seems impossible. Telling a human body to repair major trauma, severe nerve damage, or even missing appendages, literally just by focusing very specific energies onto a single point in space.
To reverse engineer exactly how this works, here's our roadmap. We'll start with building a real heal ray, then cracking the regeneration code, followed by harnessing light and sound. After that, we'll cover painting the voltage stencil, take a look inside the coaxial projector, and finally, compare healing today versus tomorrow. Section 1. Building a real heal ray and looking at the transient pulse.
Section 2. Cracking the regeneration code, moving from scar to blastema. Before we can even prompt the cells to rebuild, we have to solve a massive biological bottleneck. You see, humans already ran this incredible limb-building program once, back when we were embryos. But when we get injured now, our default response is to rapidly patch the hole with fibrotic scar tissue. Animals like the axolotl, though, do something vastly different. They create a blastema, which is basically a mass of specialized cells ready to rebuild complex structures. To mimic that axolotl and, not just grow a random horrifying blob of meat, we have to follow 4 super precise steps. First, we make the wound plastic again with a temporary pulse of specific genes, like Lin28 and MYC, which halts that scarring process. Second, we hit the biological gas pedal for outward growth using signaling proteins like Wnt and FGF. Third, we pattern it using structural regulators like BMP, so bone actually forms where bone is supposed to be. And finally, we have to tell it exactly where to build these structures in 3D space.
The absolute crucial piece of this whole puzzle is VMEM, or resting membrane voltage. This is a bioelectric stencil that can literally tell a planaria flatworm to grow 2 heads without making a single genetic edit. Discovered by the Levin lab at Tufts, this coordinate system relies on the resting membrane voltage of cells. Basically, the electrical state of the tissue acts as a structural blueprint. By just gently nudging this voltage, we can tell the body's cells build an eye here or build a limb tip here, merely by guiding the existing bioelectric circuit.
Section 3. Harnessing light and sound for non-invasive delivery.
So how do we deliver these genetic instructions without physically stabbing a cell with a needle? Well, we use sonoporation. It's basically using sound waves to politely knock the cell's door open. By vibrating injected microbubbles with a focused ultrasound phased array, we can briefly pop open 100 nanometer pores right in the cell membrane. This brilliant trick lets us non-invasively drop our transient, healing instructions directly into the cells, but strictly within the exact localized spot where that ultrasound is focused.
Section 4. Painting the voltage stencil and guiding the growth. This is where the whole recipe really comes together. Once we've nudged those cells open with sound, we need to paint that VMEM pattern we just talked about. By using pulsed electromagnetic fields, or PEMF, at very specific low frequencies, we can actually shift the cellular voltage by 10 to 30 millivolts. That is exactly what paints the bioelectric stencil required for structural growth. It's wild when you look at how it all intersects. Light acts as the remote control, giving a massive ATP energy boost while acting as an optogenetic switch to turn the growth program on. Sound safely delivers the instructions and triggers mechanosensors. And finally, electromagnetic fields paint the voltage stencil. When they all intersect at one exact point, boom, they form our quantized ball of regeneration.
Section 5. Inside the coaxial projector and how this integrates into hardware.
Knowing the biology and the physics is awesome on paper, but engineering this into a functional, physical device? That is an incredible feat of modern applied science. Think of this thing more like a chunky medical thermometer gun, definitely not a weapon. Here is exactly how the outer, middle, and center rings of the barrel are packed with safe, FDA-cleared tech. The outer ring houses that ultrasonic phased array to create the acoustic trap. The middle ring features a laser array and optics for both near-infrared healing light and high-resolution imaging.
And right smack in the center, you have the photoacoustic detector alongside the low-field PEMF coil that paints our electrical stencil. Operating this device is just a masterclass in closed-loop control. When you pull the trigger halfway, it scans the wound, meticulously mapping blood vessels and the current bioelectric state. The internal firmware rapidly calculates exactly where the beams need to intersect. Then, with a full trigger pull, it locks onto a tiny 10 to 32 millimeter voxel. Think of a voxel as a 3D pixel floating in space, exactly 15 centimeters in front of the muzzle. All three fields perfectly intersect right at that target. And the best part? Outside of that tiny dot, the beams are completely harmless and well below any bioactive threshold.
Section 6. Healing today versus tomorrow. It's time for a reality check.
Because look, you aren't going to just blast your body with this thing and magically regrow his severed arm over a long weekend. As the source material really emphasizes, this is a research concept outlining DARPA-level integration. We have to separate what this technology is capable of achieving right now from the ultimate holy grail of human regeneration. Full limb regrowth is a massive biological marathon that takes months of sequential patterning and vascularization. But the physics inside this projector can already do absolute wonders today. Accelerating bone fracture healing, closing stubborn chronic wounds, or repairing peripheral nerves at safe, FDA-cleared doses is just as life-changing for a patient right now as full limb regrowth might be tomorrow. The biology of a human arm is the bottleneck here, definitely not the optics of the gun.
To wrap up this explainer, I want to leave you with this pretty incredible thought from the source material. We aren't building a ray gun here. We're building a 3D printer for human tissue. You'd hold this projector and meticulously paint the wound, voxel by voxel, layer by layer, holding that bioelectric pattern steady until the tissue actually self-assembles. It truly makes you wonder, what limits of human biology are we going to break once this closed-loop system is fully perfected?